Hello, this is Husna and my topic is about the stages of leukocyte. So, leukocyte means what? WBCs. The leukocyte is known as the soldiers of our body. Why do we call them as the soldiers of our body? Because they defend against the foreign particles. So, the stages of leukocyte, it is a process which can be defined as a leukopoesis, which can be explained through this definition, which is leukopoesis. Leuko means leukocytes, which is WBC. Poiesis means synthesis, means the process. What does it mean? Means leukopoesis is a process of the development of the WBC, the maturation of the WBC, development as well as the maturation of the WBCs. This process is called as a leukopoesis. So, we can see the formation of the WBC in the leukopoesis. So, if we think this as a red bone marrow, where does the WBC forms? Here the leukopoesis takes place, which means the development and as well as the maturation of the WBC takes place. As we know, the blood cells like RBC, WBC and as well as platelets, they form from stem cell. The alone stem cell forms the RBC, WBC and as well as platelets. In the leukopoesis, during the formation of the development or the maturation of the WBC, we are going to see the 6 stages of the leukocytes. What are the 6 stages of the leukocytes? Myeloblast, promyelocyte, myelocyte, metamyelocyte, band form, polymorphonuclear leukocyte. Now, the basic thing we have to know is the WBC has two types of WBCs, which is granular WBC, a granular WBC. The granular WBC, further are 3 types: neutrophils, eosinophils, basionophils. Here only we are going to see these 6 stages. And the another second type of WBC is a granular WBC. The granular WBCs are further of 2 types, monocyte and lymphocyte. We are going to learn about how the WBC are going to form. That is what we call them as leukopoesis process. As we know, from the bone marrow, WBC are going to form. Now, as I told you before, that whether it may be RBC or WBC or platelets, they are made from the stem cells. From these only, these 3 are formed. So, we are going to see these 6 stages in the above diagram. First of all, the starting point is the stem cell, which is also known as hematocytoblast. These hematocytoblast further forms, it divides into the 2 stem cells. One is myeloid stem cell, another one is lymphoid stem cell. So if we go here only, the myeloid stem cell is there, no. The interleukin 3 and as well as interleukin 5, a granulocyte stimulation factor is there, no. It works on the myeloid stem cell, which forms the monoblast. These interleukin 3 and interleukin 5 act on the myeloid stem cell, further forms to monoblast, and from monoblast it forms to promonocyte. From promonocyte, you can see the nucleus is differing from here, promonocyte to monocyte. The technique to memorize the 6 stages of the leukocyte is the blast and then pro and then cell. First the blast will happen, then the pro and then cell. Now we are going to learn about the next interleukin 3, interleukin 5, granular site of stimulating factor act on the myeloid stem cell. These act on the myeloid stem cell and the process happens. The myeloblast forms. Here you can see the myeloblast as a first stage. Here the myeloblast forms when the interleukin 3, interleukin 5 and the granulocyte stimulating factor act on the or work on the myeloid stem cell. The myeloblast forms. Here the myeloblast further divided into 3. Here 3 is there. Now here 3 is there. After the myeloblast, what we are going to form is promyelocyte. Myeloblast, promyelocyte. Here now we are going to see about the promyelocyte. Myeloblast over promyelocyte. Here how can we identify in the microscope means the blue stained cell having a nucleus with the blue granules. These can be known as the basophilic promyelocyte. Basophilic promyelocyte. If the myeloblast having a red color stained with the red color granules and the nucleus in it is known as the eosinophilic promyelocyte. This is a blue stained basophilic promyelocyte, eosinophilic promyelocyte, blue stained, red stained. And then the third one is neutrophilic promyelocyte. Here we can see the red as well as blue granules in it. So we call them as a neutrophilic promyelocyte. So from these stain we can identify them whether it is a basophilic, eosinophilic, neutrophilic promyelocyte. Myeloblast over, promyelocyte over, and then these are going to next further process there. This is the second. After the promyelocyte, what does it comes? Myelocyte. Here you can see the myeloblast over, promyelocyte over, myelocyte will come. Here we can see the blue granules. The nucleus is differing here. You can see, you can identify by the nucleus, by the blue stain. This is we can call it as a basophilic myelocyte. The red color having red nucleus and as well as red stained, having something different nucleus, eosinophilic myelocyte. And the third one is having red and blue stained granules in it. These are the granules, blue, red and red and blue. Basophilic myelocyte, eosinophilic myelocyte, neutrophilic myelocyte. Blue stain, red stain, both something like pinkish. And then these basophilic myelocyte next goes to form the band cell. For the same, here from in between these two stages, they will develop and they get matured. This is only we will call them as a WBC. This is only we call as a leukopoesis. The development and the maturation of the WBC is known as a leukopoesis. So here the leukopoesis is taking place and the whole cell is formed now. From the band cell, the basophil forms. Basophil, eosinophil, neutrophil. These three are what? These three are granular WBCs of the, these three are the granular WBCs. The S-shaped nuclei having the blue granules, basophil, you can identify them as basophil, having two lobes. Here the nucleus changes, having two lobes in the microscope, having a red granules in it. This is eosinophil and WBC. This is fully matured WBC now. The stages are over. Now the WBCs are done. And the third one is neutrophil, having more than 2 lobes or 3 lobes. This is known as the neutrophil. Here the granules will be red and as well as the blue. We can see red and blue granules in it. So this is a S-shaped, U-shaped. This having a lobes. This having more than 2 to 3 lobes. So we can identify them as this. And then here the stages are over. Now here the 3 WBCs, the granular WBCs of WBC are done. They will go to the blood vessels. I will explain it afterwards. First we will see here the myeloid stem cell is there. No, here the kidney and the liver releases the thrombopoietin. So it acts on the myeloid stem cell which forms the megakaryoblast. Megakaryoblast is a big cell which further divides into the promegakaryocyte and then megakaryocyte. Afterward, the stem cell is of two types, right? Myeloid stem cell, lymphoid stem cell. So this much is done. This much part is done. Here we learnt about the stages of the leukocyte. So this is the lymphoid stem cell. From this stage the WBC is going to form. From this stage the platelets is going to form. Platelets are going to form, and then from this stage, here I will explain. The lymphoblast will come. From lymphoid stem cell, lymphoblast will come, then pro-lymphocyte come, and then lymphocyte. Here the lymphocyte is there. No, it further divides into the two cells, B lymphocyte, T lymphocyte. These two lymphocytes are formed. Now we are going to learn further deeply. Here there is a junction. This is a sinusoidal capillary. This is a junction where these granular WBCs like basophil, eosinophil, neutrophil, which are ready WBCs, they move through these junctions. So what happens? The basophil is there. No, having the granules blue. This is a basophil. The first one, the monocyte, is there. No, the monocyte, because of no granule, the macrophage process will take place, which is the eating process, and then eosinophilic. This, how can we remember them? As the two lobes having a red stained, having a granules, eosinophilic. How do they enter? And then neutrophil, they having a three lobes, having a red and blue stain. These are the neutrophils. So now here I told the megakaryotes, these are the big cells. They come here at this junction. They burst because these are large and big. No, they can't enter. So they burst and forms the small fragments which is known as the platelets. What do the platelets do? They help in the blood clotting. And then I told you about the from lymphoid stem cell, from lymphocyte, B lymphocyte and T lymphocyte is formed. Here the B lymphocyte is there, T lymphocyte is there. What does the B lymphocyte do and T lymphocyte do? Means B lymphocyte come in blood and then gets activated. Where does it get activated? From in the blood. So come in the blood and then gets activated. So what does it makes? Means it makes the plasma cells and Y-shaped antibodies. The T lymphocytes are of two types, helper T cells and cytotoxic T cells. Helper T cells help the B cells, B lymphocyte cells, to make the plasma cells, Y-shaped antibodies. And then cytotoxic T cells They help in hepatosis, which means of the program of the cell death. So the T lymphocyte, they get activated in the thymus gland and then to the blood, they enter to the blood. So this is the basic information. Thank you.